Hello everyone and welcome to the second session of NPTEL course in herbal drug technology. In this week we are seeing standardization of herbal drugs as well as herbal products and formulations. In the previous session we saw what is standardization, that is a process to ensure consistency in the product, as well as the need for standardization so that the efficacy of the product is maintained. In this particular session, we will dwell a little deeper into different methods of standardization of herbal plant material. Now, in the previous session, we saw one thing, that the herbal drug standardization can be done using different quality control methods, and this quality control methods involved organoleptic methods, macroscopy, microscopy, physical, chemical as well as biological methods. Now let's understand each of this using examples. So when we come to organoleptic methods are the methods that we sense with our five senses, and that's the reason they are called as organoleptic methods. Now this can be visually perceiving it, just by color you come to know whether it's an orange or a sweet lime, or just by a color you can come to know whether you have nut galls, which are used for tanning in leather industry. Now they may vary from grey to green. Green color indicate the freshest one, whereas grey color indicate the one on which the insect has escaped and essentially may lead to poor quality. Similarly, just to give you a simple example, you can differentiate between your well fermented teas as well as green teas, like your black color and green color. Now moving to the next parameter, it could be your odor. Now odor is an important parameter, especially for drugs which are fragrant. Take for example your spices, condiments, jeera, cumin, that is your, then you have your coriander, dhania, then you have your shah jeera, caraway. Each of this might appear little similar but have a very different aroma or different flavor. So you can evaluate them by aroma. You know whether your clove is fresh or exhausted by aroma of your clove oil which is emanating from the cloves. Same happens to your cinnamon. You know whether it's a cinnamon or it's a just ordinary tree bark coming from the aroma. Now not only that, you can use your taste senses, but beware, not every drug can be tasted, as some of this can be lethal. But some drugs which you can definitely taste, as cinchona for its bitterness, it contains quinine which has which can be called as a bitterness standard. Pungency, your chilies, your black pepper, they are known to contain alkaloids which are extensively pungent, such as your piperine and capsaicin. Astringent, when you consume your amla, you get that little tickling sensation or pickering sensation on your tongue, and your tongue feels a little tactile, that's called as your astringency, as well as sweetness in cases of your stevia as well as licorice. Now in certain cases you can just understand the drug by feeling it. Say for example, some drugs like Cassia angustifolia and acutifolia. If you see your angustifolia, they are little thicker, whereas your acutifolia are more thinner. So if you take both leaves, the thicker one is definitely your Indian variety, and that is your angustifolia. The texture also plays a vital role in mucilage containing drugs. Again in senna, you will see if it is genuine senna leaf, when you add water it becomes little slimy or soapy, whereas your other plant leaves which do not contain mucilage will not turn slimy or sloppy. So this is about the organoleptic evaluation. Moving on to the depth of the next method. The next method is macroscopy. So when I want to visually see it, and this visually seeing is also part of organoleptic, but here we classify it as different because majority of the drugs are evaluated by this method. So you can do a simple measurement. Visually I see the size of Cassia angustifolia is somewhere between 20 millimeters to 50 millimeters in length and 7 to 20 millimeters in width. Whereas if you go to see the Alexandrian variant, it is between 15 to 40, a little smaller. So basis its size, if I categorize about 50 leaves, I'll come to know that this is the average dimensions of those leaflets, and based on the leaflet size, I'll come to know whether it is the Indian senna or it is the Alexandrian senna. Macroscopically, I can also understand if the drug given to me is organized or unorganized. Meaning organized means it is cellular, that is made up of cells, organs, tissues, or it is non-cellular like your mucilages, acacia, like gums or even resins or volatile oil. They are all called as unorganized matter. Now visually I can also understand what plant part it is. Say for example roots, in case of stem or leaves or flowers or fruits, just the main appearance of this can tell me which plant part it is. And one more thing which I can definitely interpret macroscopically is there any visible disease or infestation? See, sometimes when the storage is not properly done, there is certain fungus, bacteria coming out, or the leaves start to ferment. I come to know based on the appearance, and macroscopically that has to be evaluated well in order to ensure consistency of your end product. If your raw material is substandard, infected, understand your phytoconstituents are also going to be compromised here. Moving on to the next very important method, and that's your microscopic evaluation. When you're not sure of the drug by macroscopy or by organoleptic evaluation, what you can do is maybe you can take the drug, cut a thin slice of it, put it under microscope. We call it as your transverse sections, and this transverse sections help us understand the drug. Say for example, here you have a section of a stem, that is ephedra stem. What does tell me it's a stem? The presence of pith tells me it's not going to be a root, it's definitely a stem part. And then what happens here is, basis the arrangement of my xylem, phloem as well as the fibers, I come to know that this is definitely a stem. This is definitely a xerophytic plant because I deep down am not able to see here, but it shows what are called as sunken stomata. Now epidermal peel will also give you some idea. So when you peel the epidermis of a drug, it has numerous arrangement or structures like your stomata, the shape of epidermal cells, the arrangement of trichomes, glandular and covering trichomes are all seen in your epidermal peel. Now this arrangement of stomatas and type of trichomes present can tell me what the drug is. Say for example, diacytic stomata is observed in your adulsa leaves, whereas a parasitic or a parallel cell stomata is observed in your senna leaflet. I can also do one test or a chemical test under microscope. This is if I have loaded a section in this, I can add certain chemical reagents and change in the color of this particular tissue can tell me what is where, and that is what I refer to as histochemical localization. So what I can do is, if I am checking for quinones, I can add little alkali. If I am checking for allurone grains, I can add picric acid. If I am checking for mucilage, I can add what is called as ruthenium red. If I am checking for starches or carbohydrates, I can add iodine. So where all these chemical functional groups are located, I come to know by adding this reagent, and such things are called as histochemical localization. Not only that, once I put an organized drug under microscope on the basis of arrangement of stomatas, on the basis of arrangement of tissues, I come to know whether it's a monocot or a dicot plant. Not only that, in some cases, some unique arrangements, like in the case of ephedra, you will directly be able to identify the plant under the microscope. Now this is an example of qualitative or yes no microscopy. You can also do what is called as quantitative microscopy. Now I said quantitative is more to do the measurements. So I can do the measurements, but measurements of what? Measurement of size of starch, measurement of size of calcium oxalate. I can identify the type, and that will be called as qualitative. But when I measure the size, that will be called as quantitative analysis. I can do what are called as leaf constants. So there are certain constants that are there, certain values which remain constant for a particular plant species. Now this value includes stomatal number, that is number of stomata per millimeter square of a given leaf on a particular epidermis. Now understand, because stomatas are variable. The upper epidermis, the number of stomatas are different. Lower epidermis, obviously the stomatas are more, because on the upper epidermis they will be desiccated. So in plants generally they are more on the lower epidermis. So number of stomatas per millimeter square are called as stomatal number. Number of stomatal cells per epidermal cells in a given area is called as your stomatal number. Number of vein islets. Now what are vein islets is when you see a leaf, you have veins. Now this veins subdivide further to make the vasculature available to every little tissue. Now if I just magnify this, what is going to happen is these veins are going to spread and eventually they form a network. Now this is the area of green leaf which is encircled by this network. This is like an island surrounded by sea. So I call this as one islet. So this is another islet. So another area of green tissue surrounded by vasculature. In some cases your veins go and terminate inside it, and those are called as vein terminations. So in my vasculature, how many such vein islets are there per millimeter square are called as vein islet number, and how many such terminations are there per millimeter square are called as vein termination number. Because veins are in the between layer, like your cheese sandwich, it's your cheese slice, whether you see it from top or whether you see it from bottom, your vein islet number and vein termination number are going to be consistent. They are not going to change like your stomatal number. But the next value, palisade ratio, definitely changes again. In addition to that, you can also ascertain the purity of plant or certain plant organelles or certain layers of tissues by using what is called as Lycopodium spore. Similarly, I can do physical methods. Now these physical methods are based on the physical attributes of drug. Now there are drugs like carbohydrates which imbibe moisture, acacia, so or even certain gums. So when I put it in oven at about 105 degrees Celsius, what happens is the moisture goes off and there is definitely loss of weight. We call it as loss on drying. So loss on drying is an indicator of moisture as well as volatile impurities. This also includes losses due to solvents. If you have certain extracts, if the solvents haven't evaporated properly, so you have solvents also which are going to evaporate. Then we have extractive value. So if I extract about 100 gram of drug, how much of it is going to be water soluble? How much of it is going to be alcohol soluble? So extractive value is percentage of drug that is going to dissolve in a solvent or that can get extracted in a solvent. Now this solvent can be water, it can be alcohol, or it can be even chloroform. So extractive value gives you me an indication of purity. Imagine if my drug is already extracted, uska pehle we create a chai out of it. The tea or the water soluble components have already been lost and then I sell this tea powder which is already exhausted. Next time you extract it in water, its extractive is going to decrease. So IP or the pharmacopoeia specify a limit. If it follows or if it falls below that limit, that means it's a substandard drug. Similarly, we have something like an ash value. Ash value is obtained by incinerating drugs, and after incineration we don't get the organic matter, but we get what is called as inorganic matter, which includes your materials such as minerals, soil, or even metal pieces. So if any of those are present, they contribute to the ash value, and ash value beyond a certain limit is considered to be adulteration of any of this, that is your metals, minerals, or even soil. Then you have foreign organic matter. This is generally coming from plant parts other than specified. So instead of clove buds, if I am adding clove stem or clove roots, it is called as foreign organic matter. So same plant but different plant part other than drug, or some extraneous matter which could include other plant parts, or extraneous matter which could include animals such as cockroach, rodents, or their excreta, or even urine, or even their parts such as wings, or even their eggs. They are all treated as foreign organic matter. Now in addition to that, drugs which contain saponins, they have what is called as foaming index. So it's the ability to foam. Similarly, you have solubility. So how much drug or how much gram of drug dissolves in 100 ml of solvent. Then you have refractive index. Refractive index is a very good quality control parameter, especially for volatile oils which have refractive values higher than 1.45. So, if the value falls below that, you can think of it which is adulterated with paraffin or other low quality mineral oils. You have viscosity. Certain oils such as vetiver oil are very viscous, whereas certain oils are very fluid or thin, such as your lemongrass oil. Then you have your optical activity, especially your sugars, honey, they are all optically active. So if I am adulterating my honey, the optical activity of it will change. Optical activity is basically if the substance causes rotation of plane polarized light which is passing through it. Now this is seen through a sodium lamp. So if it has the ability to rotate the plane towards right, it's called as dextrorotatory. If it is rotating the plane to left, it is called as levorotatory. In our molecules, we refer this molecule as D-glucose, L-glucose, and so on. So if it is a D molecule, it has to give you a right kind of rotation or a positive rotation. If it is a L molecule, it has to give you a left side rotation or a negative rotation. If that is not observed, then your molecule is or a extract is substandard. In addition to that, you have your parameters such as melting point for fats and waxes, boiling point for oils, and congealing point especially for plant butters. So they are fixed value, like your cocoa butter melts at body temperature 37. So if it is not melting there, that means there is some problem or some adulteration with that. In addition to that, you have chemical methods of evaluation. Now this chemical methods can also be classified as qualitative, quantitative. So qualitative method includes methods to check if the compounds are present, which includes phytochemical test, to check histochemically if those compounds are present. If you check metals are present, like you can do limit tests, there are also, but limit tests are more of what is referred to as semi-quantitative because there is a benchmark. And then you can just check by TLC as we saw in curcumin. Coming to quantitative methods where you want to quantify how much curcumin is there, you could do it by like UV or certain spectrophotometric assays. You could do it by chromatographic assays such as SPTLC, HPLC, UPLC, GC. In addition to that, for standardization, you also have to quantify impurities. This includes pesticides, this includes mucotoxins such as aflatoxins, includes your residual solvents which can pose as impurities in your drugs. The last method of standardization includes your biological method. Now this is very important because the biology of your drug, that is how your drug is acting on a system, is the main parameter and is the reason for which you are taking the drug. Now this quantitative methods include, first, your hemolysis test for saponins, which can tell how much saponins are present, since saponins have the ability to break your RBCs and release the hemoglobin. Bitterness value, say for example drugs which are extensively bitter like your Karela, that is your bitter melon, like your fenugreek, like your cinchona. More the bitterness increases, that means more amount of phytoconstituents are present. In biological methods, we also see the biological load in terms of microbial contamination, which is done by microbial assays. And most importantly, the fourth parameter is the efficacy studies. So you have to do what are called as preclinical and clinical studies of your formulation of your drug to check whether it is really efficacious. If it is not efficacious, the above all the quality control methods have no meaning, because when you are going to consume it, overall there is going to be no effect on your body if my efficacy studies fail. So this is the one which is of paramount importance, and this is done with help of animal models initially and then real human volunteers in case of clinical trial. So these are all the WHO methods of standardization. Here are few more references if you wish to learn more about this, and thank you everyone for your patient listening.